Glycolysis. Come on sugar for the breakdown.
Now the cycle's got 3 phases. 1. Carbon
Mitosis, chromosomal ride in a pro-meta anatela phase. Meiosis makes eggs and sperm. It's the same in the robin as it is in the worm.
Operons have prokaryotes, turn genes off and on.
Go inside a cell, remember what you see. Hey, it's Mr. W from learn-biology.com, and I am so thrilled to bring you this video. Over the course of this video, we're going to review a huge amount of AP Biology through the songs that I've uploaded over the years on the Science Music Videos YouTube channel. Music and rhymed lyrics are incredibly effective at encoding content that you want to learn into memory. And to help you do that, I've put together a songbook. You can download it at apbiosuccess.com/songbook. Take a minute, download the songbook, and then we'll start the music. Unit 2, Cell Structure and Function. The 3 songs that you're about to hear are going to help you review the parts of the animal and plant cells, membrane structure, and osmosis. I went into a cell to get out of the rain, and there was the gatekeeper, the cell membrane. I went into a cell, and what did I see? The mitochondria, it's the energy factory. I went into a cell and said, who drives this bus? And found myself talking to the boss, the nucleus. I went into a cell to recover from a spasm, and found myself swimming in some clear cytoplasm. I went into a nucleus to ask how to get home, and got genetic info stored in a chromosome. I went into a cell, and stretching oh so far was a thin and wavy network. It's called the ER. I went into a cell trying not to be perplexed by the packaging and sorting in the Golgi complex. I went into a cell and said, who makes proteins here? And somebody responded, it's the ribosome, my dear. I went into a cell and was feeling pretty fine, till a lysosome engulfed me and dissolved me in enzymes. I went into a cell and was feeling pretty nimble, till a centriole lassoed me, tying me up in a spindle. I went into a plant cell to see how trees get so tall, and all around the outside was a rigid cell wall. I went into a plant cell, why is it so green, I asked. Cause I make food from sunlight, said a green chloroplast. I went into a plant cell to see how plant cells store food, when a vacuole informed me that he was the storage dude. So when you go inside a cell, remember what you see. There's over 1 trillion cells in both you and me. Just sing this song if you ever feel confusion, and remember active transport is the opposite of diffusion.
If you're in a cell cytoplasm heading outside, your last stop's a barrier 8 nanometers wide. The cell membrane, our subject for today, it's as basic to life as DNA. It's selectively permeable like a border patrol of a country, it maintains control, selecting what leaves or what gains entry. It's a guard, bounce, a watchman, patrolman, or sentry, but not just a guard, it sends signals and nerves. White blood cells use their membranes to eat germs like hors d'oeuvres. Membranes have receptors, enzymes, and junctions, so what kind of structure can have all these functions? C-double-L-M-B-R-A-N-E, control and transport, selective permeability, phospholipids, carbohydrates, proteins, and cholesterol, a fluid mosaic by layer in us all. So let's take a look look at what makes up the membrane. Phospholipids rule in this domain. In fact, membrane structure emerges directly from phospholipids' chemical properties. There's a head and a tail on every phospholipid. The tail's two long-chain fatty acids bound to a glycerol. It's made to order. The tail's non-polar, hydrophobic, fears water. The head's got a phosphate. It's charged negatively, makes the head hydrophilic, plays in water happily. So tail avoids water while the head's attracted to it. When phospholipids form the membrane, that's how they do it. Cause when phospholipids into water get submerged, a phospholipid bilayer structure will emerge. The tails hang together in a water-free zone. Hear the hydrophobic moan, water leave me alone. While the heads are sticking out, touching all those H2Os. Tails in, heads out, it's how every membrane goes. Tails in, heads out in a cellular sphere. It's the bilayered basis of membranes everywhere. C-double-L-membrane, control and transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol. The fluid mosaic bilayer in us all. But a cell membrane's not just a phospholipid scene. There's cholesterol, carbohydrates, lots of proteins. In fact, membrane proteins have a presence so great they often exceed the phospholipids by weight. And all of these components are in constant motion, moving, mixing like a boiling potion, flowing like dancers in a party in Passaic. That's why the membrane's thought of as a fluid mosaic. Let's start with the proteins since they're key in this mix. Transmembrane proteins span the entire width. Typically they're ports, think of channels of conveyors for things that don't go through the lipid bilayer. Any protein embedded in the hydrophobic middle built right into the inside is considered integral, while peripheral proteins either hang on the exterior or on the cytoplasmic interior. Cholesterol keeps membranes flowing with ease. In cold it keeps the lipids moving so they don't freeze. In heat it slows the lipids down, limiting their traveling. Cholesterol keeps membranes from unraveling. Membrane carbohydrates work as markers or signs, so your immune system knows which flag your cells are flying. The blood types ABO and B and A are about the carbohydrates on red blood cell membranes. C-double-L-membrane, control and transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol. The fluid mosaic bilayer in us all. C-double-L-membrane, control and transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol. I put this yummy gummy through a science experiment that models what happens to a cell in an environment that's more watery than the cell is inside. And what I saw came as no surprise. My gummy expanded, quintupled in weight. My gummy expanded and increased so great. So listen up as Mr. W explains this. It's all about osmosis. Osmosis is the diffusion of water across a membrane or a water permeable border, diffusing like all molecules in fluid situations from higher to lower concentration. Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic to hypertonic flow. Osmosis, let's go. Let's talk about our gummy in osmotic terms. Hypotonic, hypertonic, isotonic are the words you can use to discuss water's watery diffusion. Learn them well to avoid confusion. Hypotonic means higher water concentration and relatively lower solute concentration. Hypertonic is the opposite, percent of water's less, with more solute dissolved inside, as you might guess. So let's take these terms and apply them to our gummy bear. I'm looking at the package and seeing what it says here. Each bear is mostly sugar with some other stuff mixed in, and holding it together is the protein gelatin. So when you put a gummy in a cup of H2O, it's readily apparent that the gummy is so hypertonic to the water that it's in, and the water's hypotonic to our little gummy friend. And so through osmosis, the water will diffuse into the gummy, which soon will look so huge. The mass it gains is water, which will infiltrate the gummy, which one day later is looking pretty funny. Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic to hypertonic flow. Osmosis, let's go. Next I took this freshwater plant, name is Elodia. It lives in lakes and ponds, never in the salty sea. In freshwater, Elodia cells are full and firm. It's all about osmosis, as we're gonna confirm. See, the cells are hypertonic to their watery exterior, so water will diffuse into the cell's interior, expanding the membrane, pushing it against the wall. In fact, you can't see the membrane at all. But add some salty water so the outside's hypertonic, and water leaves the hypotonic cells. It's so osmotic. Membrane leaves the wall, the cells inside the scrunch with the chloroplasm molecules all stuck together in a bunch. That's why at the grocery store they always have that mister. The water on the veggies helps to keep them crisper. The droplets on the veggies are a hypotonic brew. Osmosis moves the water in the veggies look brand new. Dried fruit and beef jerky preservation is osmotic. Their low water content makes the dry food hypertonic. So any germs and molds that fall upon them lose their water and die and don't contaminate the dry food in the larder.
Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic to hypertonic flow. Osmosis, let's go.
Animals, cells and situations hypertonic, lose their water, shrink and shrivel. Looks so sick, but an animal cell in hypotonic abode gains water and expands, eventually explodes. Cause animal cells lack a wall, of course, so nothing pushes back at the osmotic force. Water flows from hypotonic to the hypertonic cell, which bursts cause the membrane can't stop the swell. If you want to keep a frog heart outside a body beating, an isotonic fluid is what you'll be needing. Water concentration same on each side of the cell. No gain or loss of water, the cells feel so swell. And a paramecium, which lives in ponds and lakes, constantly fights osmotic water uptake. The contractile vacuoles of pumping adaptation to deal with this osmotic situation.
Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic to hypertonic flow. Osmosis, let's go. AP Bio Unit 3 is cellular energetics. We're going to divide this into two parts. The first part, we're going to do enzymes, then 3 songs about the light reactions, and then a song about the Calvin cycle.
They're the protein catalyst in every organism, enzymes. Through enzymatic action, your metabolism is driven, enzymes. In Staphylococcus, jellyfish, tarantulas, and trees, they lower activation energy. Enzymes, in you and me now, enzymes. You got them in your cells where they do cellular digestion, enzymes. You got them in your mouth and in your stomach and intestines, enzymes. The thing an enzyme acts upon is called the substrate. They fit like lock and key with complementary shape. Enzymes speed up reaction rates, enzymes. An enzyme binds its substrate at its active site, enzymes. Bound together in a complex where they snuggle so tight, enzymes. New bonds will form and break through to the active site's chemistry. Reactants become products, it's the enzyme specialty. Product gets released, enzyme repeats its action readily, enzymes. Like any molecule, an enzyme shape defines its function, enzymes. Environmental changes shape leads to malfunction, enzymes. Every enzyme has a pH where it catalyzes best. A pH change will send enzyme activity to rest. Enzyme also sensitive to easily upset, enzymes. Raw heat until a certain point increases their efficiency, enzymes. But too much heat denatures them, destroying their activity, enzymes. That's why a fever running high is a dangerous situation. All that heat can alter enzymatic conformation. Keep it 98.6 for enzyme optimization, enzymes. Enzymes in saliva will break starch into glucose, enzymes. If you lack the enzyme lactase, then you won't enjoy milk lactose, enzymes. Taste tax, galactosemia, and PKU disease caused by inherited enzyme deficiencies. Enzymes, they're what everybody needs, enzymes. Organisms like plants that photosynthesize chemically combine water with carbon dioxide, making carbohydrates and oxygen too. That's why photosynthesis should matter to you. The light reactions of photosynthesis is first phase. The output's ATP and NADPH. O2 is the byproduct, and as you can see, the inputs are water and light energy. Phase 2 is the Calvin cycle, makes carbohydrate, using up the ATP and NADPH while absorbing CO2. It's carbon fixation, photosynthesis. What a sensation.
Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charged current of electricity, converted into chemical energy.
energy. Chloroplasts are the photosynthesizing organelle. You can find them in leaves in a mesophyll cell. Inside chloroplasts is a fluid called stroma. It's true everywhere from Tokyo to Roma. The stroma bathes tiny sexual thylakoids, membrane-bound sacs with a tiny space inside. The thylakoid's membrane is loaded with protein. It's the light reaction's main scene. It also has pigments like green chlorophylls, orange carotenoids, and yellow xanthophylls. These pigments absorb light that's mostly red and blue. The green is not absorbed, it reflects back to you. Every chlorophyll has a porphyrin ring with magnesium atoms in the center of that thing. In the light reactions, that magnesium gets pinged by photons, making its electron zing. Notice the chlorophyll's tail, a hydrocarbon chain which anchors chlorophyll into the thylakoid membrane. A molecule style so pretty in the thylakoid, it makes electricity. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity, converted into chemical energy. Engelmann showed us the action spectrum. He used a prism to break the sun's light into its many different colors, shown it on an algae called Cladophora. The O2 algae makes will stimulate bacteria, which will grow on any oxygen-rich area. Bacteria loved it over blue and red, but hardly grew over green. They might as well be dead. This line of growth, it's a reflection of photosynthesis's action spectrum. Note this doesn't match exactly the absorption spectra of chlorophyll A or B. Cause the carotenoids and xanthophylls we've met absorb light frequencies the chlorophylls can get, and in the thylakoid, they all cooperate in making ATP and NADPH. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity, converted into chemical energy. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity. Electron flow in chloroplasts happens along pathways in which photosystems harvest light and send electrons on their way. A photosystem is a protein complex in the thylakoid membrane that sends electrons to an electron transport chain. The non-cyclic pathway is one way electrons flow. As photosystems harvest light, away electrons go. Electrons stream from photosystem 2 to photosystem 1, a current that's powered by the sun. Photosystem 2's antenna complex starts a photon capture. Electrons bounce in response with energetic rapture. And all around the world, from Berkeley down to Haiti, electrons find reaction center P680. There, these electrons bounce like a ball, but unlike all the times before, they rise but do not fall. A primary electron acceptor grabs them away, leaving the reaction center in an oxidized state. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity, converted into chemical energy. That electron acceptor in the thylakoid membrane sends electron over to the electron transport chain, a series of proteins that takes these electrons and uses their energy for pumping protons. From the stroma to the thylakoid space, packs positive protons into that tiny place. Like charges repel, diffusion gradients created. If a proton could diffuse out, it would be elated. But the lipid bilayer prevents proton oozing. No way there's gonna be any simple diffusing. There's only one port through which protons can travel. It's the ATP synthase channel. The ACE tells you this is an enzyme which makes ATP all the time. It has binding sites for ADP and P and channels for diffusing protons whose kinetic energy changes the binding site's conformation. Binding ADP with P, which for your information is how thylakoids make ATP, life's key form of energy. From respiration, this might feel familiar. ATP is made this way in mitochondria. Osmosis is the name. ATP production is the game. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity, converted into chemical energy. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity, converted into chemical energy.
Now back to our electrons moved by the sun, flowing from photosystem 2 to photosystem 1. They get to PS1 like a used up battery. The transport chain used up their energy, but when light hits PS1's antenna, electrons bounce to reaction center. This one's P700. That wavelength makes electrons go. PS1's electron scepter grabs them away, leaving P700 in an oxidized way, and P700 with its oxidized blues accepts the electron from PS2. Back to electron from PS1. It now goes for a ride, having its own fun. But powering proton pumps ain't its fate. It travels to NADP plus reductase, an enzyme chloroplast count upon, to pass some HN electrons to NADP plus, which gets reduced. It's how NADPH gets produced. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity, converted into chemical energy. O2 is the byproduct of the light reactions, oxygen production. Here's how it happens. Remember back in PS2 it came to a rise. Reaction center P680 got oxidized. To replace lost electrons, P680 liberates electrons from water, which dissociates into one oxygen and protons too. The single O will meet another, forming O2. These protons are formed in the thylakoid space, increasing proton concentration in that place. So we see a side effect of water's oxidation is enhancement of ATP creation. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity, converted into chemical energy. Non-cyclic flow is also called the Z scheme. Put Z on its side and see what I mean. Electrons rise in energy, then fall and rise again, making ATP and NADPH, my friend. A variation on this is cyclic flow, which makes ATP, no NADPH or O. Cyclic flow results from the Calvin cycle's needs. It uses less NADPH and ATP. NADPH buildup has the repercussion of inhibiting NADP plus reduction, because NADPH clogs up reductase, keeping NADP plus from moving into place. So electrons don't go to NADP plus, they're clogged, stuck like a pimple's pus. Where can they go from P700? Nature evolved them a place to flow. A shunt pathway moves them to the ETC. I'm talking about the one in PS2, you see. These energized electrons flow and release energy, which as you know is used to synthesize ATP. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge current of electricity, converted into chemical energy. Light reactions happening in leaves, making oxygen, NADPH and ATP through a solar charge.
Calvin cycle's got 3 phases. 1. Carbon fixation. 2. Invest energy and harvest G3P. 3. Regenerate RUBP.
It's time to learn the Calvin cycle. In this recital, we're going to learn the story of a process that's so vital, cause it's how the matter making up the biosphere got here in every plant and animal you see. Yeah, it happens in a chloroplast, enzymes so fast, fixing CO2 within the stroma to a mass. Carbs like glucose, fructose, and even starchy amylose, some of which will pass from plants to you and me.
Calvin cycle's got 3 phases. 1. Carbon fixation. 2. Invest energy and harvest G3P. 3. Regenerate RUBP.
The process has 3 phases. First is carbon fixation. An enzyme called Rubisco combines CO2 with 5 carbon. RUBP stands for ribulose bisphosphate. The 6 carbon product immediately disintegrates, leaving us with 2 3-carbon molecules, setting up conditions for Calvin cycle phase 2. Enzymes take these molecules of carbons 3 and give them energy from NADPH and ATP. NADPH and ATP were generated in the light reactions now they're used to make the Calvin cycle happen. Makes glyceraldehyde 3-phosphate G3B, the harvestable product used for carbohydrate energy.
Calvin cycle's got 3 phases. 1. Carbon fixation. 2. Invest energy and harvest G3P. 3. Regenerate RUBP.
Now on to phase 3, regenerate RUBP. An enzymatic team takes 5 G3Ps, reorganizing them, which requires ATP, synthesizing 3 5-carbon RUBPs. We're at the cycle's end, but also its commencement. Rubisco's primed to work with RUBP as a catalyst. Rubisco, it's in chloroplasts and ginkgo species, roses, beeches, ferns, pines, peonies, and all the algae in the seas. Rubisco, it's the carbon fixer. It's the reason why you're here. It brings the carbon in your ear, toes, hair, and derriere into our biosphere. Don't underestimate Rubisco's worth. It's the most abundant protein on planet Earth.
Calvin cycle's got 3 phases. 1. Carbon fixation. 2. Invest energy and harvest G3P. 3.
Regenerate.
REVP. Calvin cycle's got 3 phases. 1. Carbon fixation. 2. Invest energy and harvest G3P. 3.
Regenerate.
REVP.
And now part 2 of Unit 3 with 3 songs about cellular respiration, glycolysis, the Krebs cycle, and the electron transport chain.
Hey, sugar. Come on over here, sweetie. Let's break it down. Glycolysis is a series of reactions, enzymatic actions, energy transactions. Takes glucose, a molecule so sugary, breaks it down for NADH and ATP. It's an anaerobic cytoplasmic pathway that amazes. Organized easily into 3 phases: investment, cleavage, and energy harvest. Tell me later which one you like best. Investment, activation, energy supplied. Cleavage, our 6 carbon sugar divides. Harvest, we get our energy yield. So beautiful, so intricate, keep your eyes peeled. Glycolysis, come on sugar for the breakdown, for the breakdown. Investments, like striking a match, that energy you put in makes the fire catch. For glycolysis investments, 2 ATPs, which act as activation energy. Enzymes take phosphates from ATPs, jam them on a glucose, rearranging it to fructose, leaving fructose 1,6-bisphosphate on the table with 2 phosphates. It's highly unstable, moving us to the 2nd phase, the cleaving of fructose bisphosphate, because glycolysis means splitting sugar, you can see. Cleavage yields 2 molecules with carbons 3. One is glyceraldehyde 3-phosphate G3P. It continues on our pathway, broken down for energy, but the second one, an enzyme, will immediately convert into a second G3P. Glycolysis, come on sugar for the breakdown, for the breakdown. Phase 3, G3P gets rearranged and oxidized by an enzymatic assembly line that harvests energy from each G3P, 1 NADH and 2 ATPs. Double this yield per G3P to 2 NADH and 4 ATPs. That's the gross yield for every glucose in. A generous accounting of glycolysis' win, but 2 ATPs were invested in phase 1, so you net just 2 you can use to jump or run. Put 2 in, get 4 out, your net gain is 2 ATPs that you can use. If that doesn't seem like very much, it's 'cause it ain't. There's tons of energy left in pyruvate. The 2-3 carbon molecules we're left with at the end, and what happens to pyruvate's gonna depend on the metabolic pathway where pyruvate gets sent. If it's anaerobic, it'll be fermented, but in aerobic cells, pyruvate's termination will be the Krebs cycle and total oxidation. Glycolysis, come on sugar for the breakdown, for the breakdown. Glycolysis starts with investment of 2 ATPs to the glucose that we started with. The product is cleaved into 2 G3Ps from which the cell harvests NADH and ATPs. This anaerobic pathway is respiration's first phase, billions of years old, evolved in ancient days before O2 accumulated in the seas, before eukaryotic cells arrived on the scene. It's everywhere, ubiquitous in every organism. Bacteria, sequoia tree, no matter your metabolism. Happens in the cytoplasm, doesn't need no organelles. You want to find glycolysis? Look in any cell. Glycolysis, come on sugar for the breakdown, for the breakdown. Glycolysis, come on sugar for the breakdown. Matrix of the mitochondria of all our cells is the cycle of reactions that won Hans Krebs a Nobel Prize. The cycle takes the energy and food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADH, which later brings electrons to the electron transport chain. And Krebs makes FADH do its function is the same. Krebs also makes some ATP, another claim to fame. We're talking Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2. Right before the cycle's a transitional part, links Krebs to glycolysis so Krebs can start. Enzymes rake a CO2 off a pyruvate. yields an NADPH and acetyl-CoA. Acetyl-CoA carries carbons too, with all the energy that derives from food. It's a highly reduced molecule with energized electrons that fuels up the Krebs cycle as it cycles on. Now at the start of Krebs, this acetyl-CoA has the 2 carbons it carries in ripped away. Enzymes put these on oxaloacetate with carbons 4, make 6 carbon citric acid. Who could ask for more? Notice 3 carboxyl groups on that citric acid. That's why the cycle's also named for tricarboxylic acid, or TCA Cyclic. It's acronyms that you prefer. Krebs in honor of its discoverer, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADPH, 1 FADH2. Enzymes work on citric acid and remove the CO2, and other enzymes modify and oxidize it too. The 5-carbon result is alpha-ketoglutarate. Krebs cycle, it's so great. Every oxidation can power the reduction of NAD+, which gains electron carrying function, become an NADH, that energy sensation, which later on in respiration powers ATP creation. Another oxidation follows another CO2 removed, leaving us with a 4-carbon molecule. Another NADH results from this oxidation. As Krebs does its energy transformations, this 4-carbon molecule, acetyl-CoA, still has lots of energy enzymes can take away. A series of reactions yield 1 ATP, the cell's main energy currency, leaving just enough energy for enzymes to reduce an FADH to FADH2, and 1 last NAD+ will also get reduced as the final electron carrier NADH gets produced. We've harvested what energy came in at Krebs' start. Now we have oxaloacetate at this final part. Oxaloacetate is the commencement and finale, ready to meet acetyl-CoA, and here at the final tally. Krebs goes round and round, such an ancient cycle, spinning like the wheels of my bicycle. Krebs is like the axle of aerobic respiration. I breathe out its CO2 with every exhalation. We've walked through the cycle, so now let's review. Input is acetyl-CoA with carbons too. The carbons get removed, releasing CO2. Exhaling sends the CO2 out of you. The cycle's functions: energy transformation, 3 NADH, 1 FADH2 creation, and also synthesis of 1 ATP, which cells directly utilize for energy. For every glucose cells absorbed, the cycle runs 2 times. As long as cells get fuel, Krebs is running just fine. It precedes electron transport chain, it follows glycolysis. It's spinning round and round in the mitochondrial matrix, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2.
Welcome to the story about cell energy. The goal is explaining how cells make ATP. It happens in the mitochondria, which you can think of as the cell's energy factory. Mitochondria are double membrane organelles, an inner membrane and an outer one as well. The mitochondrial matrix is the fluid inside. It's where reactions like Krebs cycle reside. Glycolysis and Krebs make NADH and FADH2 from energy and food. These electron carriers make their way from the matrix to the inner membrane. And that's where you find the electron transport chain. It's a series of enzymes embedded in the membrane, which take the electron carriers' electrons and uses their energy for pumping protons. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusing protons' kinetic energy to make ATP from ADP and P. The chain is a series of enzymes in a row. Each accepts electrons and lets them go to the next carrier in this transport chain. It's kind of organized like a bucket brigade. What drives electrons down this enzymatic series is the growing level of each carriage electronegativity into oxygen. Electrons ultimately fall. It's the most electronegative of them all. It's NADPH that starts this run, donating electrons to complex number 1. This powers active transport as protons are displaced and get pumped from the matrix to the intermembrane space. From complex 1, electrons flow to ubiquinone, also known as Q, which floats through the inner membrane happily and brings its electrons to complex 3. And you can guess the function of complex 3. It's another proton pump using electron energy. Protons jam up in that intermembrane compartment like 100 people in a one-bedroom apartment. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusing protons' kinetic energy to make ATP from ADP and P. Complex 2 is for FADH2, which donates electrons, which then get passed to Q, which once again passes them to complex 3, which pumps protons using electron energy. From complex 3, the electrons proceed to another mobile carrier, cytochrome C, which donates the electrons to complex 4. Another proton pump, could you ask for more? After complex 4, electrons flow to oxygen, which is ever so electronegative. It pulls electrons down the chain, keeping them moving like the cars of a train. And NaSO2 does this electron grabbing trick. It also grabs protons from the matrix. They all combine to form H2O. Electron transport chain, watch it go. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusing protons' kinetic energy to make ATP from ADP and P. Now all these protons in the intermembrane space are trapped. They can't get out of that place, cause protons are charged and could never get through a phospholipid bilayer. They can't diffuse, but like all particles, they're dying to go from where the concentration's high to where it's low. Stuck in the intermembrane space, they're frustrated to diffuse to the matrix. They're highly motivated, and this gradient's been made steeper by O2, which absorbs protons from the matrix stew. So from proton pumping and oxygen's actions, add another force, electrochemical attraction. Think of all those trapped protons, each one's positive. The matrix in comparison is negative. Opposites attract, so the protons are dying to get to the matrix. Oh, how they're trying. There's only one channel that lets protons pass, and they use it like high school students busting out of class. It's a channel and an enzyme. It's ATP synthase, the closer in this game, an energy ace. ATP synthase is embedded in the inner membrane. How it works is so cool, it's insane. It's got channels for diffusing protons running right through it. When cells make ATP, well, watch how they do it. The matrix side of ATP synthase has binding sites for ADP and P, which come in and bind. And as ATP synthase lets protons barge through, their kinetic energy gets put to use. Like water through a turbine, proton movement generates rotation, changing synthases, binding site conformation, which catalyzes chemical bond formation. ADP and P make ATP that energy sensation. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusing protons' kinetic energy to make ATP from ADP and P. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place.
If you're studying for the AP Bio exam, I want to let you know about a great resource that I have for you. It's a checklist that enables you to assess what you know and what you don't know. You can download it at apbiosuccess.com/checklist. Now let's move on to Unit 4, where I have songs about cell communication and mitosis.
Have you ever had a sudden fright? Perceived a threat, you're all geared up for fight or flight. Heart pounding, face flushed, hair standing up on end. It's partly from a hormone called epinephrine. Epinephrine's got another name, adrenaline. Secreted from adrenal glands, it's a stuff in EpiPens, a shot can save your life. If If you have an allergy involving sensitivity to nuts distinct from bees, epinephrine's polar, so it can't diffuse through the lipid bilayer. So instead, it induces a second messenger, it's cyclic AMP, which stimulates massive cellular activity. Let's learn how liver cells respond to an epinephrine dose. What they do is break down glycogen to glucose, which streams into your blood to give you energy required to respond to threats as you fight or flee. How does epinephrine get your liver cell to do this function? It's 3 steps: first reception and transduction, then cellular response and enzyme activation. We'll learn about the process of cell communication. Cellular communication works through phases 3. Reception of the ligand, the complementary, second messenger released, transduction is a beast. Now amplified response until the stimulus has ceased. Today's focus is G protein coupled receptors. We'll leave the other systems for another lecture. Epinephrine's the ligand that with the receptor binds. It's a hormone, the hydrophilic kind. Ligands bind to receptors only if the shape matches its specific, based on complementary attachment. Epinephrine goes everywhere but only affects cells with receptors with which it can connect. Once epinephrine binds at the receptor binding site, the receptor changes shape on its cytoplasmic side, or just inside the membrane you'll find the G protein, which until epinephrine binds is dormant, asleep. In its dormant form, the G protein's bound to GDP, but epinephrine binding makes it bind with GTP. Then the G protein drifts in the membrane space till it bumps into adenylyl cyclase, a membrane-bound enzyme whose signaling functions activating a second messenger, setting up transduction. Transduction converts a message into another kind. Message 1 was a ligand, which came along to bind with the receptor, which in response woke up the G protein, which gets adenylyl cyclase to take an ATP and remove 2 phosphates, making cyclic AMP. That's the second messenger. It takes a message deep into the cytoplasm where it unleashes a chain of enzyme action, a phosphorylation cascade. The message was received, it was then transduced. Next we'll see how cellular response gets produced. Cellular communication works through phases 3. Reception of the ligand, the complementary, second messenger released, transduction is a beast. Now amplified response until the stimulus has ceased. A phosphorylation cascade involves a chain of enzymes known as kinases. These kinases get phosphorylated, which means gaining a phosphate, which activates them. And what do kinases do when they're charged up with a phosphate? They find the chain 6 kinase and phosphorylated. This chain of activation is like falling dominoes, because that's how a phosphorylation cascade goes. And each kinase carries out multiple activations, causing massive signal amplification. The signal grows in strength with every kinase action, building in intensity, a chain reaction. One epinephrine binds, causing many cyclic AMPs, each of which activates many kinase proteins. By the chain's last kinase, tens of thousands activated. A cellular tsunami as the chain is terminated. Glycogen phosphorylase is the enzyme at the end that makes glucose by hydrolyzing glycogen. So into the blood from the liver, glucose gets secreted, and the threat that started the story can be fled from or defeated. It's adaptive how your cells are poised to spring into action. Once they hear epinephrine's alarm, there's a reaction in all eukaryotes, from amoebas to acacias. G protein coupled receptors used in cell communication. Cellular communication works through phases 3. Reception of the ligand, the complementary, second messenger released, transduction is a beast. Now amplified response until the stimulus has ceased. Back to the ligand of the receptor. It only stays for a moment before it diffuses away. So when the thread is gone, the cascade gets shut down. With no adrenal secretion, the receptor's unbound. G protein drops the phosphate, now bound to GDP. It goes back to sleep, stops its activity. Bound to GDP, it no longer stimulates adenylyl cyclase, which no longer creates cyclic AMP, so the second message stops. Kinase phosphorylation quickly drops as other enzymes, protein phosphatases, clip off phosphates, turn off kinases. Glycogen phosphorylase stops hydrolyzing glycogen, so blood glucose normalizes. Liver cells return to their resting state. I love how G proteins let cells communicate. If you like this song, please subscribe. Your comments and likes help our channel thrive. And check out our website, Learn-Biology. I wrote it for you. And there you'll see tutorials and quizzes, AP reviews, and our app Biomania. You can use it to study for AP Biology. You can get a 4 or 5, that's our guarantee. Cellular communication works through phases 3. Reception of the ligand. Guys, I want everyone's attention. We're going to go over what we did yesterday. We're going to go over the stages of mitosis. All right, everybody ready? All right, let's go. Interphase of cell division's longest part. Nuclear membranes intact as it starts. The cell's growing, cytoplasm flowing, chromosomes get duplicated, DNA gets replicated, chromosomes are spread out so they can't be seen distinctly, but note the nucleolus, the ribosome factory outside the nucleus, the two centrosomes. They later make a spindle which will pull apart the chromosomes. Prophase follows, the chromosomes condense, each is made of two sister chromatids like an X. Each sister is a clone, the closest of kin, and a centromere connects them like Siamese twins. The nucleolus disappears, it melts away, as the cell takes a ribosome production holiday. The centrosome separates, starts spindle formation for separating chromatids and cell elongation. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In late prophase, pro-metaphase, the nuclear membrane disintegrates. The centrosomes migrate to the cell's opposing sides, and between them the fibers of the spindle wend and wind. The spindle's made of microtubule fibers which attach to chromosomes. A kinetic causes a protein patch that serves like a handle that the fibers can grasp. When they pull apart the chromosomes, splitting them in half, the spindle moves the chromosomes with nudges so fine into linear formation on the 50-yard line, a location equatorial, defined in metaphase, where the chromosomes are lined up in that middle place. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. The spindle fibers pull on the kinetic cores. A cellular, molecular, mitotic tug of war. The centromere snaps, sisters get separated. Now these chromatids are chromosomes. They've been upgraded. The snap and separation defines anaphase. The A for apartness, for moving different ways. Kinetic core, spindle fibers separate. The sisters see I'm waving goodbye, calling out I'm gonna miss you. And the other spindle fibers push and grapple like felons, make the cell elliptical like a watermelon. In telophase, membranes form around the chromosomes that spread out as a nucleoli come on home. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In animal cells, there's a ring of microfilaments that form at the equator, and they cinch themselves in tighter till the cell is in two pieces. Yeah, in animals, that's cytokinesis, but it's different in plants, and then the cell divides by building a new cell wall from the inside as the Golgi sends vesicles with cellulose goo, which makes a plate, then a wall divides a cell in two. And instead of one mother cell, we now have daughters too. Identical twins, kinda old but kinda new. From your single cell beginning, this is how you grew. And for single celled eukaryotes, it's reproductive too. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Hey guys, eukaryotes go.
Your success in AP Biology starts here. Are you struggling with AP Bio? With learn-biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. The AP Bio exam is coming up, so go to learn-biology.com/student sign up to find out how learn-biology.com can help you crush the AP Bio test and master AP Biology. For AP Bio Unit 5, I've got my song Meiosis, and then right after, you're going to get a special treat.
Well, Max, you think they're ready for meiosis?
Ready for whose osis?
Meiosis, let me show ya. Meiosis is how we make sex cells or gametes. The sperm cells or egg cells performing the feat of moving genes forward in eukaryotes like orchids and bees. Meiosis doesn't happen in all cells of the body. There's just a few cells that have this hobby. I'm talking about germ cells and testes and ovaries. Germ cells are diploid, and what that means is that the chromosomes are paired up in teams. In each pair, one's from your dad, one's from your mother. When you line up the chromosomes, it's suddenly clear how each is a member of a coupled pair. Homologous pairs, each a homologue of the other. In humans, the diploid number is 46, and a key trick that happens in meiosis is dividing that number in half to 23. And that single set of chromosomes has its own name. It's called haploid in this meiotic game. So diploid to haploid's a key meiotic strategy. Meiosis makes eggs and sperm. It's the same in the robin as it is in the worm. Makes haploid gametes recombination. Meiosis creates variation. In interphase 1, meiosis starts. It's the DNA replication part, an evolutionary relic of its origin. Cause meiosis evolved from mitosis, you see, so each process starts identically, replicating chromosomes into two sister chromatids. In prophase 1, chromosomes coil up and homologous pairs pair up, forming tetrads, each with chromatids 4. A chiasm is the spot where the chromatids link, and synapsis is a name for the whole darn thing, and crossing over is what this whole process is for. See, the homologs aren't identical twins, no way, they're not the same DNA. The genes are the same, but the alleles might take different forms. So during synapsis, alleles can cross over between homologs, and when it's all over, there are gene combinations that have never been seen before. Meiosis makes eggs and sperm, it's the same in the robin as it is in the worm. Makes haploid gametes, recombination, meiosis creates variation. Another meiotic variety creator is metaphase 1 with homologs at the equator, because how each pair lines up is random and independent. So when one pair facing north might be the maternal, and the next one it might be the paternal. It's a 1 and 2 shot, it's called independent assortment. So 2 pairs divide up in 4 distinct ways. It's 2 to the number of pairs, you could say. So think about us humans with 23 homologous pairs. 2 to the 23rd power is a number so great it's 8,388,608. That's why metaphase 1 is a variety creating affair. And now connect this assortment with recombination. Note that what we've got during gamete creation is sperm and egg cells that are absolutely unique. So if you ever wondered why sisters or brothers can be so different from one another, just remember these meiotic recombining techniques. And if meiosis had never evolved, the book of life would be a very different tome. 'Cause if it wasn't for meiosis, all offspring would be clones. Metaphase 1, homologs to the equator. Anaphase 1, they say see you later. It's like mom and dad splitting up and setting up new homes. 2 nuclei form in telophase 1, then cytokinesis. Meiosis 1 is done. We've got 2 haploid daughters still with doubled chromosomes. Now things are much simpler in part 2 of meiosis. Essentially it's just like mitosis. You just need to pull those sister chromatids apart. They line up in the center in metaphase 2. Anaphase pulls them apart, we're almost through. Telophase, then cytokinesis, we're at the last part. Meiosis makes egg and sperm, it's the same in the robin as it is in the worm. Makes haploid gametes, recombination, meiosis creates variation. Well, Max, how do you think we did? You think they understand meiosis now?
You know, I don't know why anyone would ever want to understand Eurosis.
Max, it's not Eurosis, it's meiosis. Haven't you been listening to the song?
I know it's not Meiosis, that's why I said Eurosis.
Max, if you don't understand meiosis, you're never going to be able to pass your genes on.
You know what, I'm going to keep my genes on, and I want you to keep Eurosis to yourself.
AP Bio Unit 5 is about meiosis and genetics, and I have a song about Gregor Mendel that I've played for my students over the years, but I've never recorded. I thought I'd play it for you. Here we go. Mendel, Gregor Mendel, he's a big name in biology. His work in genetics says a lot about heredity. Mendel said that alleles segregate and independent assortment is just great. Mendel, Gregor Mendel, he's a big name in genetics, and in genetics, a man before his time. AP Bio Unit 6 is gene expression, molecular genetics, and I've got songs about DNA structure and function, DNA replication, protein synthesis, operons, bacterial gene regulation, viruses, and cloning.
Welcome, I'm so happy you came by for a lesson about the essence of biology. Why DNA is a topic that's so fantastic. We're talking about deoxyribonucleic acid. If you've ever wondered about development in elephants and bumblebees, coffee trees, well, DNA's the recipe. In its home, in a chromosome in the nucleus, DNA drives the bus genetically controlling us. In ferns and worms, infectious germs, mommy's egg and daddy's sperm, DNA's the information linking up the generations. It's a code of life, so listen up well, cause DNA's the molecule in charge of every cell. DNA, fantastic, deoxyribonucleic acid. We're talking about deoxyribonucleic acid. DNA, fantastic. The shape is key, probably well known to you. Double helix, twisted ladder, or a double corkscrew. Imagine a twisted length of tape with deoxyribose sugars on the edges with phosphate. Sugar, phosphate, making up the sides of the ladder with nitrogenous bases on the inside. With the bases like the rungs, together with deoxyribose sugar and a phosphate make a nucleotide. Deoxyribonucleotides are the monomers, link them together for the DNA polymer. 0.34 nanometers marks the space between one nucleotide and the next one's place. And for the helix to make one turn takes 10 bases in a row, as you can discern. Since space between nucleotides is 0.34, one turn takes nanometers, 3.4. So stylish, so slick, first described in 53 by Watson and Crick. And don't forget Franklin, she took that famous X-ray pic, but she died, so she never got a Nobel Prize. DNA, fantastic, deoxyribonucleic acid. We're talking about deoxyribonucleic acid. DNA, fantastic. The nitrogenous bases come in one of four varieties, known by the letters A, T, C, and G. A is for adenine, thymine's got the T. C, it stands for cytosine, guanine's got the G. A and G with two nitrogen rings are purine. C and T with one ring are pyrimidines. C only bonds with G, A only with T, because like puzzle pieces, they're complementary. They fit together, snuggled up like hand and glove, like enzymes and substrates, like people in love. A, T, C, G matched up like lock and key, forming hydrogen bonds, either 2 or 3. Hydrogen bonds, you know they aren't very strong, but they're enough to hold DNA together all day long. The way the bases fit together couldn't be sweeter. They give DNA a width of 2 nanometers. DNA's double-stranded, looks pretty swell. The strand's orientation is anti-parallel, with one strand standing up, the other on its head. It's how they fit together, Crick and Watson said. DNA, fantastic, deoxyribonucleic acid. We're talking about deoxyribonucleic acid. DNA, fantastic. Sugars and phosphates make the backbone, the structure, but the sequence of bases is where you find instructions for development of bodies in the cytoplasmic symphony that constitutes like miracle on goats and golden algae. Your sequence of bases is unique, a special batch. Unless you're an identical twin, it's nowhere matched. And you can bet that if you're not Django Fett, that you've got no clone. No one like you's been known. Our bases comprise our individuality. You might have an A where I have a G, explaining why I'm bald and you got all that hair. These differences can show up anywhere. The protein hemoglobin, listen up well, it carries oxygen in your red blood cells. Hemoglobin's made of 4 protein pieces, a trait widely shared among animal species. Hundreds of bases spell one hemoglobin piece, hundreds of A's, G's, T's, and C's. The gene starts CACG, TGCAC, then TGAGGA, CTC, CTC. The key is these bases are information for hemoglobin's function and conformation. Hundreds of bases in a predetermined order. A single change brings on a major disorder. Change T to A in one single spot. This little point mutation might not seem like a lot. Thymine to adenine might not seem that big to you, but baby, that's the cause of sickle cell anemia. DNA, fantastic, deoxyribonucleic acid. We're talking about deoxyribonucleic acid. DNA, fantastic. DNA, fantastic, deoxyribonucleic acid. We're talking about deoxyribonucleic acid. DNA structure with its bases complementary makes replication easy, but not quite elementary, since A only bonds with T and C with G. The double helix seems to copy naturally, or as Crick and Watson said, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. You first unzip the DNA in one or more places, breaking hydrogen bonds to separate the bases. Each resulting single strand serves as a template, allowing enzymes to replicate new strands with complementary bases that match. And through hydrogen bonds, these bases attach. Each nucleotide now bonds to the next through a sugar phosphate bond they connect. Meiosis and style proven 58 that this is how the double helix replicates. One strand new, the parent strand preserved. In other words, the whole thing is semi-conserved. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Now let's see how replication really goes with blind, mindless enzymes controlling the show. Made more complex by something you can see. Each DNA strand has directionality. 5' to 3 is how the enzymes go. Just refer to the carbons in deoxyribose. So when a new strand is synthesized, nucleotides get added on the 3' side. The process begins with helicase, which opens up the helix at a special place, breaking hydrogen bonds at the origin. A sequence telling helicase where to begin. A replication fork is now composed, where both parent strands have their bases exposed. And to keep the double helix from rewinding, single-strand proteins come in and start binding. No two forks always form when DNA doubles. The whole thing's called a replication bubble. Now it's primase's turn, the next enzyme, to come to the origin at this time. Primase lays down a primer of RNA, complementary to the template DNA, setting the stage for the star of our show. DNA polymerase now set to go. DNA polymerase's job is to add deoxyribonucleotides to a growing strand, but polymerase needs a growing strand in place, which is why initiation is the job of primase. What happens now is simple, it's a replication race. As polymerase follows helicase, as the fork opens up, replication proceeds with nucleotides added at incredible speed. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Said applies to the leading strand, where replication smooth, continuous and grand. But on the second strand, fork opens 3 to 5, a direction where polymerase can polymerize. So instead of following helicase, polymerase moves away from the fork in place. So replication's lagging and fragmentary, as discovered in '66 by Okazaki. So the lagging DNA is filled with Okazaki fragments and RNA primers, and to clean up this mess, polymerase 1 removes the primer, puts the deoxyribonucleotides in what could be finer. And now the fragments need to be connected so the new DNA can be perfected. Ligase carries out this function with pride, sealing sugar phosphate bonds between nucleotides. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Look at what's coming out of the nucleus, destined for a ribosomal strand of mRNA. It's got the code, the information for the protein we'll be making today. The RNA message is organized into codons. That's a sequence of bases 3 that spell out one and only one amino acid, a genetic code that works universally. A codon has its match in an anticodon, 3 bases on tRNA's bottom side. tRNA's job is to bring amino acids to ribosomes so proteins can be synthesized. mRNA, tRNA, and ribosomes plus amino acids make a protein making machine. Read the codon, make a polypeptide, that's translation. Cells synthesize proteins. The small subunit of a ribosome binds with an mRNA's leading end and slides until it reaches star codon AUG, indicating where translation begins. A ribosome has 3 binding sites where tRNAs can bind. The A site's first piece, next then E, and the E's got an exit side. The first tRNA in this translation process binds a start codon and binding site P, and that tRNA carries that amino acid that goes by the name of methionine. And now, to finish this initiation, the ribosome needs to be made complete. The large subunit binds a small subunit. Now we're ready for translation to proceed. mRNA, tRNA, and ribosomes plus amino acids make a protein-making machine. Read the codon, make a polypeptide, that's translation. Cells synthesize proteins. Now we're ready for the next step, elongation. An amino acid carried by a new tRNA with an anticodon matching with the second codon binds at the site called A. Then the ribosome carries out an enzymatic action binding these amino acids with a peptide bond, so a dipeptide hangs on the A site tRNA. Watch translation move along. Next, the ribosome shifts exactly 1 codon over in a move that we call translocation. So the A site's empty and the P site's loaded, and the first tRNA's in the E location. E site tRNA makes its exit, and the next tRNA fills a site called A, and the ribosome catalyzes away. In your cells it happens every second, every day. And watch that polypeptide, see it get longer as each codon gets translated. Ribosomal robots reading RNA instructions as proteins get created. mRNA, tRNA, and ribosomes plus amino acids make a protein, make a machine. Read the codon, make a polypeptide, that translation. Cells synthesize proteins. Protein synthesis, translation. Protein synthesis, translation. Protein synthesis, translation. Protein synthesis, translation. We started with initiation, which was followed by part 2 of translation, elongation. Now it's time to finish synthesizing this protein with a finale called termination. When the ribosome gets to a stop codon, there's no tRNA anticodon that matches, so A site's bound by a release factor, the polypeptide detaches. tRNAs and P and E sites float away, ribosome breaks apart, no longer one, the polypeptide folds into a functional protein. Translation is done. mRNA, tRNA, and ribosomes plus amino acids make a protein, make a machine. Read the codon, make a polypeptide, that translation. Cells synthesize proteins. mRNA, tRNA, and ribosomes plus amino acids make a protein, make a machine. Read the codon, make a polypeptide, that translation. Cells synthesize proteins. Protein synthesis, translation. Protein synthesis, translation. Protein synthesis. Hey, I'm E. coli, a bacterial cell. Gonna tell you about something I do so well. It's the way I turn my genes off and on. It's a regulatory system called an operon. My chromosome has over 4 million bases and 4,000 genes, so I need some bases for deciding which gene gets activated. And operons are how my genes are regulated. One operon has initials TRP, the trip operon. Its function, you see, is coding for enzymes that make tryptophan, an amino acid. Here's my plan. If there's tryptophan around, then E. coli shouldn't make it, so I shut the system down. But if tryptophan is lacking and it's something I require, I turn the system on and make what I desire. But how does the bacterium ascertain what and when to synthesize when it's got no brain? Let's examine the structure to get the information about the workings of operonic gene regulation. Operons have prokaryotes, turn genes off and on. A cluster of genes transcribed as a single RNA. Operons have prokaryotes, turn genes off and on. Repressing, inducing the operonic way. Let's start with the structural genes, which code for enzymes and other proteins. And these structural genes get transcribed as one big RNA transcript. We've just begun. These structural genes are under the control of one promoter. The promoter's role is to let RNA polymerase bind and read the structural genes and transcribe. After the promoter's an operator that controls if the genes are transcribed now or later. The operator's where a repressor protein binds. If repressor's attached, nothing transcribed. The repressor's the product of a regulatory gene located away from this structural scene. The repressor has 2 binding sites. It functions like a switch for turning on a light. Site 1's for tryptophan. When it's occupied, the repressor binds the operator on the other side. But when site 1's empty, site 2's confirmation won't bind the operator's location. So when tryptophan is in the environment, it binds with the repressor and makes the other side stick to the operator. Blocking transcription, the repressor's on a regulatory mission. Think of the logic, the energy saved. If tryptophan's around, E. coli shouldn't make it. But if tryptophan's something E. coli can't find, the repressor changes shape and no longer binds. So RNA polymerase transcribes the genes to make tryptophan that I require. When I've made enough, the tryptophan binds and affects the repressor, which changes shape and now connects to the operator. Blocking transcription, its negative feedback evolved to perfection and adaptation. Such efficiency. Come on, class, sing about operons with me. Operons have prokaryotes, turn genes off and on. A cluster of genes transcribed as a single RNA. Operons have prokaryotes, turn genes off and on. Repressing, inducing the operonic way. The trip operons, the repressible kind. Now let's learn about the inducible kind. This one's called lac. It involves lactose, a disaccharide in milk, as everyone knows. Lac structural genes make proteins and enzymes for hydrolyzing lactose to monosaccharides. But if lactose is absent, I shut this down, cause I only need these enzymes when lactose is around. Lac's the opposite of trip, who we just met. The default conditions offer the repressor sitting on the operator, blocking transcription, so no enzymes are made for lactose digestion. When lactose is around, it diffuses into me and binds with the repressor, which shifts beautifully into a form where it can't bind the operator. Transcription starts like an escalator. See how lactose is an inducer, inducing transcription. What a brilliant maneuver. Then these enzymes break lactose down until it's digested and no longer around. And when it's gone, there's no lactose to bind with the repressor, which shape shifts on the other side and binds the operator, so transcription ends. It's negative feedback again. Operons have prokaryotes, turn genes off and on. The cluster of genes transcribed as a single RNA. Operons have prokaryotes, turn genes off and on. Repressing, inducing the operonic way.
If you like this song, would you please subscribe and please leave me a comment or a question? Try out my website, Learn Biology, and if you're open to another suggestion, download my app. It's called Biomania from the App Store and Google Play. With my website and app, I guarantee a 4 or 5 on the AP exam this May.
I wanna clone, I don't wanna be alone. I wanna talk to a younger me on the phone. I wanna sing harmony with a younger me in the same key. I want a new companion who's the closest of kin, a 50-year younger identical twin. I'm going to the cloning lab to make myself a copy of me. Back in '62, Sir John Gurdon took the nucleus from a body cell of an adult frog, poked it in an egg cell that had its nucleus removed. This fused cell grew to be a polywog. That polywog was a clone of the adult, the first scientifically created animal clone. And 50 years later, the Nobel Prize Committee made $1 million call in Mr. Gurdon's home. We've all seen clones before, identical twins. The clones, two separate people with the same DNA. Or if you've ever made a cutting from a plant, you've done some cloning, but not quite in Mr. Gurdon's way. He took a single cell from the body of an adult and from it made a being genetically the same. A brand new approach to animal reproduction, a huge shift from Mother Nature's game. So if I want to clone, I'm heading over to the lab where they'll extract a nucleus from a somatic cell. That's any cell that's not for sex. It could be bone or gut or muscle, or my epidermal cells could do as well. Cause all these cells, despite their different look and feel, possess all of the genes required to make a new you or me. And this idea that's key to cloning underlies the principle that all cells have genetic equivalency. I wanna clone, I don't wanna be alone. I wanna talk to a younger me on the phone. I wanna sing harmony with a younger me in the same key. I want a new companion who's the closest of kin, a 50-year younger identical twin. I'm going to the cloning lab to make myself a copy of me. Next I'll get an egg cell, which I can of course provide, and we need to ensure it has no genes inside. Cause my clone's a clone of me, I want fidelity and nothing besides. One way we can enucleate the egg is to irradiate it, shine some UV light on it, which ruins the DNA in it. Now put my nucleus in it so that the DNA in it is mine and only my genes that to my new clone I will transmit. And now electric zap provides so that the cell starts to divide, no longer just my zygote clone, it starts to be an embryo. It needs a place to grow, so to a surrogate mom it will go. And 9 months later, my clone's new life begins. Though half a century apart, identical twins will be on Letterman and Leno, and we'll fly to San Francisco, and everywhere we go we'll be the center of the show. I want a clone, I don't want to be alone. I want to talk to a younger me on the phone. I want to sing harmony with a younger me in the same key. I
want a new companion who's the closest of kin, a 50-year younger identical twin. I'm going to the cloning lab to make myself a copy of me. This sounds like just a story. Remember all the glory. Ian Wilmot got a 96 for cloning the sheep dolly, and then another volley. Genetically modified Polly, every single cell she had contained a human gene, oh golly. In years that followed, camels, deer and ferrets, mice and cows and hogs, pet owners have paid $50K to clone their cats and dogs, and even rhesus monkeys, which are primates just like we are. It's completely doable. You see, the techniques come so far, but now I'm here inside the lab. I'm starting to think, maybe not. My clone, of course, would be a person, not my minion or a robot. And what if my genes don't activate correctly and there's problems? And instead of singing harmony, a sickbed's where my clone will be, and I'll be right there by his side, sick with regret, not feeling pride. My narcissistic grandiosity had got the best of me. My self-absorbed preoccupation with genetic duplication simply has no place in a world with 7 billion population. I don't need a clone. It's okay to be alone. When I need someone, I'll talk to a friend on the phone. I wanna sing harmony with the people who I find around me. Come back soon when I follow this lesson with a song about regenerative medicine, a way that we can clone that brings some benefit to humanity. And just a tiny preview of the kind of thing we might consider. Imagine if for some reason you needed a new liver. Instead of finding donors and running the risk of rejection, we clone your cells and grow you a liver that suits you to perfection. I want a clone. I don't want to be alone. I want to talk to a younger me on the phone. I want to sing harmony with a younger me in the same key. I want a new companion who's the closest of kin, a 50-year younger identical twin. I'm going to the cloning lab to make myself a copy of me. I'm a virus, an infectious particle, an anathugger pirate, the genuine article, Ebola, chickenpox, West Nile, influenza, yellow fever, AIDS, herpes, SARS are coming right at me. I'm not a cell, not an independent organism, don't even have my own metabolism. I only reproduce myself by taking over cells, then I bust them apart, the one that you reveal well. My structure is simple, mostly proteins and genes, packaged as a sub-micron killing machine. My capsule's made of capsules and self-assemble. Whenever cells see me, they start to tremble. Inside my capsid is where my genes reside. I got DNA or RNA on the inside, and the rest of me is various proteins. Whose role is assisting in assault cells and taking control. I'm a virus, controls your cells to replicate. I'm a virus, controls your cells to replicate. The Lytic cycle shows one method of attack. I attach to the self, surface piercing like a tack. Inject my genes inside, use my victim machinery to make my genes and proteins to reproduce some more of me. Step 1, tail 5 is grab the membrane or the wall. 2, inject my genes, the cell is gonna fall. 3, use the victim, polymerase and ribosomes to synthesize my proteins and replicate my genome. 4, tail piece and capsule self-assembly. 5, I'm all together now, look at all my progeny. 6 is lysis, I burst the cell apart. Now I'm looking for more victims I can heal and depart. I'm a virus, controls your cells to replicate. I'm a virus, sometimes instead of lysine I lysogenize, which happens after I inject my genes inside. I slip into your chromosome, deep inside your womb. A menacing and silent presence, bringer of doom. Integrated in you, your DNA's my home. And see what happens when you replicate your chromosomes. Each copy that you copy copies me as well as you. You might make a million copies, there's nothing you can do. My lurking hiding beings call it Trophic or virus. And if you got another name, just call us a virus. Or write it on papyrus till the moment I emerge from your DNA and Lytic. Now a self-destroying surge, and suddenly your cells are dying. Watch me as I lies them. I'm a pirate, so forget your plans. The program now denies them. Your cells are virus factories and making millions more of me. I lose your cells and now I'm free infecting. Use my destiny. I'm a virus, controls your cells to replicate. I'm a virus, now meet my associate, the virus HRV.
whose helpless target is a cell called T. HIV causes immunodeficiency, raising cancer and infection vulnerability. Remember that the T is an essential cell in the immune system army, it's the general. So knocking out the T cell sets the stage for cancer germs and viruses like me to invade. HIV's genes are made of RNA. Outside its capsids, a phospholipid membrane, a skin stone from the cell last dispatched, studded with proteins that landed attached to a protein on the T cell called CD4, inducing that cell to open up its door, inviting in HIV's enzymes and pour the T cells already done for. HIV is a retrovirus, its sex routine has to do with transforming its RNA genes. Reverse transcriptase takes its RNA and reverse transcribes it into DNA. Then this DNA integrates into your chromosomes like a lysogenic prophage in your genome. This proviral DNA is transcribed and translated as new virus genes and proteins get created. And like antivirus, all those particles self-assemble into new viruses, which of course resemble the original particle that started the infection, acquired by sex or a needle injection.
Virus, control cells to replicate. Virus, the natural cells to replicate. Virus, control cells to replicate.
AP Bio Unit 7 is about evolution. The College Board's name for this unit is natural selection. Not a good name because evolution is about so much more than natural selection. But the one song that I've written is about natural selection. It's about the peppered moth. Enjoy it.
Meet the peppered moths of the English woods. The forest of Manchester is its neighborhood. It's gone from white to black and back through natural selection from bird attacks. Natural selection starts with inherited variation, followed by selection of favored traits within a population. If selected pressures consistently applied in the same direction, the beneficial traits will become the norm. It's how adaptations form. Peppered moths spend their days on the trunks of trees. A Manchester bird will eat each moth it sees. Protective coloration is a key adaptive trait. A moth that stands out meets a painful fate. In 1800 that meant looking light, 'cause lichens on tree bark made them look so white. So the light-colored moths in the population had a phenotype to avoid predation. Natural selection starts with inherited variation, followed by selection of favored traits within a population. If selected pressures consistently applied in the same direction, the beneficial traits will become the norm. Is how adaptations form. Through the moth gene pool, white genes had spread. Genes for black were rare, their owners wound up dead. Then industrial pollution turned things head to foot. The lichen whitened trees covered with black soot. Those rare black moths started to thrive. Instead of getting eaten every year, they survived. As the white moths were eaten one by one, the black ones bred. It was oh so fun. Each moth generation takes a year, so soon the shift was very clear. The dark phenotype came to predominate, and a young white moth met a painful fate. Natural selection starts with inherited variation, followed by selection of favored traits within a population. If selected pressures consistently applied in the same direction, the beneficial traits will become the norm. Is how adaptations form. About 1960, things turned the other way. Humans realized pollution doesn't pay. With less soot in the air, the lichens came back. The tree bark in Manchester turned to white instead of black. I'm sure you can guess what ensued. The change turned the black moths into bird food. Today, Manchester moths are mostly white, as the birds keep on selecting, eating all black moths in sight. Natural selection starts with inherited variation, followed by selection of favored traits within a population. If selected pressures consistently applied in the same direction, the beneficial traits will become the norm. Is how adaptations form.
In the 1950s, Kettlewell proved a moth with poor camouflage becomes bird food. When Kettlewell's critics created a fuss, the mechanism was confirmed by Meiosis. AP Bio Unit 8 is Ecology, and I've got two songs for you. The first one is the carbon cycle, and the second is about food chains and trophic levels. Carbon atom in a CO2 up in the air, in the air, in the air. Floating with the oxygen and nitrogen up there, nitrogen and oxygen up there. And there I'm gonna be until I'm sucked into a tree, into the leaves of a tree. Photosynthesis captures me, puts me in the leaves of a tree. Fixed to the leaves, I'm in a new situation. Now I'm a solid in a plant location. Cause photosynthesis does carbon fixation, carbon fixation. Carbon cycle moving me around, gaseous in the air and solid on the ground. Carbon cycle going round and round, going round and round and round. I might be fixing to a tree or herb or weed or shrubbery, or maybe an algae floating in the sea. But after CO2 enough produces where I'll be, producers where I'll be. Formerly a gas, now I'm a solid carbohydrate. Leaves of rock and fruit and wood are mostly carbohydrates. Or possibly a grain of rice on someone's dinner plate, someone's dinner plate. Then maybe I'll be eaten by a deer or mouse or bird, chewed by a goat or by a buffalo herd. After being swallowed I could be drugged as a turd, dropped down as a turd. Carbon cycle moving me around, gaseous in the air and solid on the ground. Carbon cycle going round and round, going round and round and round. In a plant or animal, I might get respired. For energy, I'll get respired. CO2's released and oxygen's required. Oxygen's required. Or possibly it carries me before fall to the ground. Onto the soil on the ground. Decomposers break me down and CO2 comes out. CO2 comes out. Decomposition, respiration put me in the air. Carbon dioxide in the air. With the clouds of nitrogen and oxygen up there. Nitrogen and oxygen up there. Carbon cycle moving me around. Gaseous in the air and solid on the ground. Carbon cycle going round and round. Going round and round and round. Or maybe I fall to the ground, no oxygen around. Logs and leaves accumulate pound by pound. Makes me into fossil fuels deep under the ground. Deep under the ground. In mind the coal and oil and gas combust me in machines. CO2 emitted from machines. Now the CO2's at levels no one's ever seen. No one's ever seen. Carbon cycle moving me around. Gaseous in the air and solid on the ground. Carbon cycle going round and round. Going round and round and round. Carbon cycle moving me around. Gaseous in the air and solid on the ground. Carbon cycle going round and round. Going round and round and round. Oh, a nuclear reaction happens deep inside the sun, where hydrogen atoms are fused into helium. Then their heat and light is radiated into space, and 90 million miles it shines upon the Earth's face. For every 100 calories that shines up from the sun, the calories are green plus triple number only one. But every single calorie in ocean, hills, and plains will serve as a foundation for a living food chain. At the bottom of the food chain are producers like green plants, whose cells make food from sunlight, making use of chloroplasts. It happens in the pines and flowers on your windowsill, cause every single green plant's got a little chlorophyll. Well, the next step in a food chain is a primary consumer, an herbivore, a cow, or vegetarian baby boomer. A primary consumer spends its mealtime eating plants. It might be a buffalo or it might be an ant. Each level in a food chain yields only 10% of the former level's energy. You might as swear it will. There are bones and leaves you can't digest that really make a dent. And don't ignore the weight and cost of metabolic rent. So if your plants are yielding about 100 calories, your yield will only be 10 in the birdies and the bees, and 700 calories in algae in the sea. Primary consumers, it will yield you 70.
Some other ways of getting food are really out of sight. You could be a predator or be a parasite. A predator eats meat, a parasite eats your insides, and if you're a decomposer, you will leave me when I die. And if you know this about food chains, you can really take the cake. When energy goes through, it doesn't last, it dissipates. The nutrients in food chains meet a very different fate. They recycle and they circulate forever, ain't that great? Just remember there's no beings in this world that live alone. Populations form communities where all do make their home. Though your niche may be determined in the role that you are playing, every creature alive or dead is in a food chain.
This musical review of AP Biology has been so fun. Thank you for joining me. If you want to learn more, do two things. First, sign up for learn-biology.com, and second, watch this next video.